Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
What if I told you that the very building blocks that make up our bodies, those tiny little amino acids, could actually be helping cancer cells hide from our immune system? Kind of a scary thought, right?
It really is. And that's exactly what we're diving into today. Some pretty groundbreaking research that shows how a common amino acid, arginine, is linked to the sneaky ways breast cancer cells can evade those all-important immune defenses.
It's like cancer has figured out how to exploit even the most basic processes in our bodies to its advantage.
Absolutely.
And we're not just speculating here. We've got this brand new study hot off the press from the journal Cancer Cell by Zhu and their team. Plus, we've got some health news perspectives to help us put it all into context. So in this deep dive, we're going to take all this complex science and break it down for you, our listener.
Exactly.
We want to understand how this whole arginine thing works and what it might mean for developing new cancer treatments down the road. No need to get bogged down in all the super technical details. We're going to give you the key takeaways. So let's start with the basics. What were these researchers trying to figure out in the first place?
Well, they were really interested in understanding how arginine metabolism, basically how cells use and process arginine, plays a role in the growth of tumors.
Okay, so arginine and tumors. Got it.
Right. And they were particularly focused on what was happening within the tumor microenvironment.
The tumor microenvironment. So
That's basically the immediate surroundings of the tumor, all the different cells and molecules interacting in that space.
Okay, makes sense.
They wanted to pinpoint which cell type, by interacting with cancer cells through arginine, had the biggest impact on how the cancer grew and spread.
So they were looking for the key player in this whole arginine tumor connection.
Exactly.
And did they start by looking at actual patients?
They did. They analyzed blood samples from a pretty large group of women, 608 to be exact, who all had breast cancer.
Wow, 608. That's a pretty big study group.
It is. And what they found was pretty striking. They saw a connection between how advanced the cancer was, looking at things like the size of the tumor, whether it had spread to the lymph nodes, and whether it had metastasized to other parts of the body, and the levels of arginine in the patient's blood.
So the more advanced the cancer, the higher the arginine levels.
Exactly. And here's the interesting part. Out of all the amino acids they looked at, arginine had the strongest link to cancer progression.
So that's a pretty big red flag right there.
It is. And it's like the first clue in a detective novel. Something's definitely going on with this arginine and how it relates to cancer getting worse.
Makes you want to keep reading, right? So where was all this arginine coming from? Like, what was the source of it in these tumors?
Good question. To figure that out, they used a really cool technique called single-cell RNA sequencing.
Single-cell RNA sequencing? That sounds pretty high-tech.
It is. It allows you to look at the activity of genes in individual cells, so you can see which genes are turned on and off in each cell type within the tumor.
So it's like getting a personalized readout of what each cell is up to.
Exactly. And their analysis showed that the cancer epithelial cells, those are the cells that actually make up the bulk of the tumor, were the primary producers of arginine within the tumor microenvironment.
So the cancer cells themselves were churning out arginine.
Yep, particularly a subtype called basal cells.
Basal cells. Now those are?
They're a type of epithelial cell that tend to be more aggressive and associated with certain types of breast cancer.
And why were these basal cells pumping out so much arginine?
Well, they had really high levels of an enzyme called arginosuccinate synthetase 1, or ASS1 for short.
ASS1, got it. And this ASS1, it basically helps make arginine, right?
Exactly. It's a key enzyme in the arginine production pathway.
And they did some fancy calculations, right, to confirm this whole arginine source thing.
They did. They took into account both the levels of ASS1 activity and the number of each cell type present, and they estimated that nearly half of the arginine in the tumor microenvironment was being generated by those cancer cells.
Wow, almost half. That's significant.
It is, and it suggests that the cancer cells are going out of their way to produce arginine.
And did they find any connection between this ASS1 enzyme and how advanced the cancer was in those patients they looked at initially?
Actually, they did. They used immunohistochemistry, which is a technique that lets you visualize specific proteins in tissue samples.
Visualize proteins, so they could actually see this ASS1 in the tumor samples.
Yep. And what they saw was that tumors from patients with more advanced cancer had significantly higher levels of ASS1 compared to tumors from patients with earlier stages of cancer.
So more ASS1, more advanced cancer, Another red flag.
It is. They also found a direct correlation between the amount of ASS1 and the levels of arginine in the patient's blood.
So more ASS1 means more arginine being produced, which we already know is linked to worse outcomes.
Right, and here's the kicker. High levels of ASS1 were associated with shorter time to metastasis.
Meaning the cancer spread to other parts of the body more quickly.
Exactly. And this was especially true for patients with basal-like tumors and those where the cancer had already spread to the lymph nodes.
So the more arginine those cancer cells are making, the more aggressive the cancer seems to be.
It's looking that way.
Okay, so we've got cancer cells making a lot of arginine, and that's linked to bad news for the patients. But is this arginine actually causing the cancer to become more aggressive, or is it just a byproduct of something else that's going on?
That's the million-dollar question. To really nail down cause and effect, they moved on to experiments in living organisms.
You mean like animal studies?
Exactly. They used mice as models for breast cancer. They started with mice that had normal functioning immune systems and gave them a type of breast cancer called 4T1.
4T1, okay.
What they found was that when they gave those mice extra arginine, the tumors grew faster.
So just pumping them full of arginine made the cancer worse?
It did. But here's the flip side. When they used techniques to knock down ASS1 in the cancer cells, basically reducing the amount of arginine those cells could produce,
the tumors grew more slowly.
Exactly.
So it really seems like arginine is playing an active role in this whole process.
It does. But then they did something really interesting. They repeated these experiments, but this time they used mice that didn't have fully developed immune systems.
So their immune systems couldn't fight off the cancer as effectively.
Right. And guess what happened? The effects of both giving extra arginine and knocking down ASS1 completely disappeared.
Really? So it didn't matter if they had more or less arginine, the tumors grew the same way.
Exactly. And that points to something really crucial. The immune system is playing a critical role in how arginine is affecting tumor growth.
So arginine isn't directly fueling the cancer cells themselves. It's acting through the immune system.
That's a really important distinction. And they saw the same results when they used another type of mouse breast cancer model, EO771.
Okay, so arginine is up to something sneaky with the immune system. But which immune cells are involved in this whole scheme?
They went back to their single-cell RNA sequencing data to figure that out. They were looking specifically at the interactions between different cell types within the tumor microenvironment and how arginine metabolism played a role.
Like a cellular detective story.
Exactly. And both in the human and mouse breast cancers, they found the strongest arginine-related interaction was between cancer cells and macrophages.
Macrophages. Now those are...
They're a type of immune cell that can either fight against cancer or, unfortunately, sometimes actually help it grow.
So they can play for both teams.
Yeah, kind of like double agents. And what's interesting is that these macrophages had very low levels of ASS1 themselves.
Meaning they couldn't make their own arginine.
Right. So they were likely getting it from somewhere else. And guess who was conveniently supplying it?
The cancer cells.
There you go.
So the cancer cells are like the arginine dealers in this scenario, and the macrophages are their customers.
It's a pretty good analogy.
And what does this arginine do to these macrophages? Like, how does it change their behavior?
Well, they did some experiments where they treated macrophages in the lab with arginine, and they found that it increased the production of certain signaling molecules called cytokines.
Cytokines. Remind me what those are again.
They're basically chemical messengers that cells use to communicate with each other.
Okay, so the arginine was making the macrophages send out different signals?
Right. And specifically, it increased the production of cytokines that promote tumor growth and suppress the immune response.
So bad news all around.
Yeah. These cytokines, things like TGF-beta1, CD274, which is also known as PD-L1 and IL-10, they all help the tumor thrive and prevent the immune system from doing its job.
So the arginine is essentially turning these macrophages into traitors.
It is. It's pushing them towards a pro-tumor state, what's called an M2-like state.
M2.
It's a way to classify different types of macrophages based on their function. M2 macrophages are generally associated with wound healing and tissue repair, but in the context of cancer, they can actually help the tumor grow and spread.
So it's like the cancer is hijacking a normal process and using it to its advantage.
Exactly. And when they looked at the macrophages that had taken up arginine from the cancer cells, they found that these macrophages were expressing more genes related to this M2-like state.
So the arginine was actually changing the way their genes were being expressed.
It was. And conversely, when they limited the amount of arginine available to the macrophages, they saw the opposite effect. The macrophages started expressing more genes associated with an anti-tumor or M1-like state.
M1 being the good guys in this scenario.
Right. M1 macrophages are the ones that attack and destroy cancer cells.
And did they see any evidence of these different macrophage types in the actual tumors?
They did. They looked at the tumors from those mouse models, and they found more of these M2-like tumor-associated macrophages in the control tumors compared to the tumors where they had knocked down ASS1 in the cancer cells.
So less arginine being produced by the cancer cells meant fewer of those traitorous macrophages.
Exactly. They also looked at human breast cancer samples and found a marker for M2 macrophages, called CD163, was present at higher levels in more advanced cancers, and those levels correlated with the amount of arginine in the patient's blood.
So it all coming together.
It is.
Cancer cells make arginine. Arginine turns macrophages into tumor helpers. And the more of those tumor-helping macrophages you have, the worse the prognosis.
It's a pretty grim picture.
It is. But hold on. We've got to know for sure that those macrophages are really essential for this whole cancer growth scheme. Like, what if they're just bystanders and something else is driving the process?
That's a good point. To really confirm the role of macrophages, they did some experiments using a special type of mouse called an NOG mouse.
NOG mouse, what's that?
It's a type of mouse that's immunodeficient.
Meaning?
It lacks a fully functioning immune system.
Yeah.
And importantly, these NOG mice can accept transplants of human cells without rejecting them.
So they can be used to study human cells in a living organism.
Exactly. So what they did was they transplanted human breast cancer cells into these NOG mice.
Okay.
Some of the cancer cells had normal levels of ASS1, and some had been modified to have reduced ASS1 levels.
So some were pumping out lots of arginine and some were making less.
Right. And they did this transplantation in a couple different ways. In some mice, they just transplanted the cancer cells alone.
Okay.
And in other mice, they transplanted the cancer cells along with human immune cells called PBMCs.
PBMCs, that's
Peripheral blood mononuclear cells.
right. And those include macrophages.
They do. So they had these two groups of mice, some with just cancer cells and some with cancer cells plus PBMCs, and they were watching how the tumors grew.
Got it.
And here's where it gets really interesting. They found that knocking down ASS1 in the cancer cells only slowed down tumor growth in the mice that also received the human PBMCs.
So if the mice didn't have those extra immune cells, including macrophages, reducing arginine production didn't make much of a difference?
That's right. It suggests that those immune cells, particularly macrophages, are playing a crucial role in this whole process.
But they didn't stop there, did they?
They didn't. They wanted to be absolutely sure that it was the macrophages that were making the difference. So they did one more experiment. They took the human PBMCs and specifically removed the macrophages from them before transplanting them into the mice.
So no macrophages this time.
Right. And what they found was that when they did that, the tumor growth inhibition they had seen before completely disappeared.
Wow. So without those macrophages, it didn't matter if the cancer cells were making more or less arginine, the tumors grew at the same rate.
Exactly. And that proves that macrophages are absolutely essential for this effect to occur.
So the cancer cells are making arginine, the macrophages are taking it up, and that's somehow changing the macrophage's behavior to help the tumor grow. But what's the actual mechanism? Like, what's happening at the molecular level?
They did some more lab experiments to figure that out. They used what's called conditioned media, which is basically the liquid that cancer cells have been growing in.
So it's full of all the stuff the cancer cells are secreting.
Right, including arginine. They took this conditioned media and treated a type of macrophage called M0 macrophages with it.
M0, what are those?
They're kind of like the precursor cells that can differentiate into either M1 or M2 macrophages.
So they're not committed to either side yet.
Right, they're kind of like the undecided voters in the immune system. And what they found was that treating these M0 macrophages with the cancer cell conditioned media caused them to polarize or differentiate into more of an M2-like state.
So they were being pushed towards the dark side.
Basically. And when they analyzed these M2-like macrophages, they found altered levels of arginine and another molecule called ornithine.
Ornithine, that's it.
It's a molecule that's involved in the production of polyamines.
Polyamines, those are.
They're molecules that are important for a lot of cellular processes, including cell growth and proliferation.
Okay, so arginine and ornithine, and we're talking about polyamines. What's the connection?
Well, it turns out that arginine is actually a precursor for polyamine synthesis.
So the arginine that the cancer cells are making is being used by the macrophages to make polyamines.
Exactly. And the altered levels of arginine and ornithine they found in the M2-like macrophages suggested that this pathway was more active in these cells.
Meaning they were churning out more polyamines.
Right. And they confirmed this using a technique called isotope tracing, where they could actually track the fate of specific molecules within the cells.
So they could literally see the arginine being converted into polyamines.
They could. And they also found that the macrophages weren't able to make arginine themselves from a related molecule called citrulline.
So they were totally dependent on the cancer cells for their arginine supply.
Exactly. It's like the cancer cells have created this whole system where they control the macrophage's metabolism.
And when they knocked down ASS1 in the cancer cells, what happened?
It reduced the amount of arginine the cancer cells were secreting.
So less arginine for the macrophages.
Right. And that led to a decrease in the expression of those M2 macrophage markers we talked about earlier. CD163, TGF-beta1, IL-10, and PDL1.
So basically the macrophages were less pro-tumor.
Exactly. And importantly, they could restore the expression of those markers by adding arginine back into the culture.
So it really does seem like arginine is the key to this whole manipulation scheme.
It's a pretty compelling case.
Okay, so we've got the cancer cells secreting arginine, the macrophage is taking it up, converting it into polyamines, and becoming more pro-tumor. But how does this actually affect the immune system's ability to fight the cancer? Like, how do these pro-tumor macrophages interfere with the good guys?
Well, they did some experiments where they co-cultured these M2-like macrophages with CD8 plus T cells.
CD8 plus T cells, those are
Those are the killer T cells, the ones that are responsible for directly attacking and destroying cancer cells.
the special forces of the immune system.
Exactly. And what they found was that the M2-like macrophages actually suppressed the activation of these killer T cells.
So they were preventing them from doing their job.
They were. They measured the expression of various markers on the T cells, and they found that the T cells that had been co-cultured with the pro-tumor macrophages had reduced levels of activation markers like CD25, granzyme B, interferon gamma, and TNF-alpha.
So they weren't getting the signals they needed to go into attack mode.
Right. And they also had increased levels of exhaustion markers like PD1 and TM3.
Exhaustion markers, what are those?
They're basically signs that the T cells are getting worn out and losing their ability to fight.
So the macrophages were not only preventing them from activating, but they were also tiring them out.
Exactly. And this suppression of T cell activity was less pronounced when they knocked down ASS1 in the cancer cells.
So less arginine, less suppression.
Right. And they saw something similar when they looked at their single-cell RNA sequencing data from human breast cancers. They found an enrichment of exhausted CD8 plus T cells and a reduction in activated CD8 plus T cells in tumors that had high levels of ASS1.
So in tumors where the cancer cells were making a lot of arginine, the killer T cells were more likely to be exhausted and less likely to be activated.
Exactly. It all fits together.
It's like the cancer cells are setting up this whole elaborate system to disarm the immune system.
That's a good way to put it. They're not just hiding. They're actively creating a hostile environment for the immune cells that are trying to fight them.
And a key part of that strategy is manipulating arginine metabolism to turn macrophages into their accomplices.
Precise.
So it's not just the cancer cells themselves that we need to worry about, it's the whole ecosystem within the tumor.
Absolutely. And understanding how that ecosystem works is crucial for developing new and more effective treatments.
Okay, so we've got cancer cells producing arginine, macrophages taking it up, converting it into polyamines, and becoming pro-tumor, which then leads to the suppression of killer T cells. It's a pretty complicated story, but it all makes sense.
It does.
But you mentioned polyamines earlier. Can you remind me again why those are so important in this whole process?
Sure. So arginine is the starting material for polyamine biosynthesis, and the researchers found that the levels of various polyamines like ornithine, putrescine, spermidine, and spermine were all increased in the M2-like macrophages.
So the macrophages were churning out more of these polyamines.
They were. And they also saw increased activity of the enzymes that are responsible for making these polyamines, like ARG2, ODC1, and SMS.
ARG2, ODC1, and SMS.
Arginase 2, ornithine decarboxylase 1, and spermine synthase.
Right, the polyamine making crew.
Exactly. And using that isotope tracing technique again, they confirmed that the arginine the macrophages were taking up from the cancer cells was indeed being used to produce these polyamines.
So it's a direct link.
It is.
And were all of these polyamines equally important in this process, or was there one that stood out as being particularly influential?
That's a good question. They did some experiments to test the effects of each individual polyamine on the macrophages, and they found that spermine was the most potent in driving that pro-tumor shift.
Spermine, so that's the main culprit.
It seems to be. When they treated macrophages with spermone, it was the most effective at increasing the production of pro-tumor cytokines and suppressing the production of anti-tumor cytokines.
So spermine was really turning up the volume on those pro-tumor signals.
It was. And they also found that the conditioned media from spermine-treated macrophages was even better at inhibiting the activation of CD8 plus T cells compared to the media from untreated macrophages.
So spermine was making those macrophages even more effective at shutting down the killer T cells.
Exactly. It really highlighted the central role of spermine in this whole process.
Okay, so spermine is bad news. But how is it actually doing all this? Like, how is it changing the macrophage's behavior at a molecular level?
Yeah, they dug even deeper, and they discovered that spermine was influencing something called epigenetic modifications in the macrophages.
Epigenetic modifications, those are...
They're changes to the DNA that don't alter the actual genetic code itself, but they can affect how genes are expressed.
So they're like switches that can turn genes on or off.
Exactly.
Yeah.
And one of the most common epigenetic modifications is DNA methylation.
DNA methylation.
It's basically the addition of a methyl group to a DNA molecule, and it can silence gene expression.
So if a gene is methylated, it's less likely to be expressed.
Right. And what they found was that spermine was causing a decrease in DNA methylation in the macrophages.
So it was essentially taking the brakes off of certain genes.
Exactly. They did some detailed analysis of the methylation patterns in the macrophages, and they found a significant number of genes that had become less methylated in the M2-like macrophages compared to the M0 macrophages.
So spermine was causing widespread changes in gene expression.
It was. And to confirm that this was really important for the pro-tumor effects, they used a drug that inhibits DNA demethylation.
So they were preventing those epigenetic changes from happening.
Right, and guess what happened? It led to a decrease in the production of those pro-tumor cytokines, and they also saw a reduction in the number of CD163 plus macrophages.
So blocking those epigenetic changes really did make a difference.
It did. It shows that these spermine-induced changes in DNA methylation are crucial for the pro-tumor effects.
And did they pinpoint any specific genes that were being affected by this change in methylation?
They did. One of the key genes they identified was PPRG.
PPRG.
That stands for peroxisome proliferator activated receptor gamma.
Okay, and what does PPRG do?
It's a transcription factor, which means it controls the expression of other genes, and it's known to play a key role in regulating macrophage polarization.
So it can influence whether macrophages become M1 or M2.
Exactly. And they found that treating macrophages with spermine led to reduced DNA methylation at the PPR gene, which in turn led to increased expression of PPRG.
So spermine was turning up the volume on PPRG.
It was. And remember that drug that inhibits DNA demethylation. They found that treating macrophages with that drug actually reversed the spermine-induced increase in PPRG expression.
So it really was the changes in methylation that were driving the increase in PPRG.
Exactly. And to really nail down the role of PPRG, they used a drug that specifically blocks PPRG activity.
So they were shutting down PPRG directly.
Right. And that suppressed the spermine-induced production of pro-tumor cytokines, and it also reduced the ability of the macrophages to suppress CD8 plus T cell activation.
So PPRG really does seem to be a key mediator of spermine's effects.
It does.
Okay, so we've got spermine reducing DNA methylation, which increases PPRG activity, and that's what makes the macrophages more pro-tumor. But what's actually doing the demethylation? Like, what enzyme is responsible for removing those methyl groups from the DNA?
They identified an enzyme called TDG.
TDG.
It stands for thiamine DNA glycosylase.
Okay, and what does TDG do?
It's an enzyme that's involved in DNA repair, and it can actually remove certain types of modified bases from DNA, including methylated cytosine.
So it's like a DNA editing tool.
It is. And they found that the expression and activity of TDG were both increased in macrophages that had been treated with spermine or the cancer cell conditioned media.
So spermine was boosting TDG activity.
Exactly. And to confirm that TDG was really important for this whole process, they knocked it down in the macrophages.
So they reduced the amount of TDG?
Right. And that inhibited the macrophage's shift towards a pro-tumor state. And it also reduced their ability to suppress T-cell activation.
So TDG is definitely playing a key role in this pathway.
It is.
But how does Spermine actually increase TDG activity? Like, what's the connection there?
They found that Spermine was activating a signaling pathway in the macrophages that involves a protein called P53.
P53, that's a familiar name.
It is. It's a tumor suppressor protein that's involved in regulating cell growth and death.
So it's usually a good guy.
It is.
Eh.
But in this case, it seems to be playing a more complex role. They found that treating macrophages with Spermine increased the expression of genes that are typically activated by P53, and it decreased the expression of genes that are normally suppressed by P53.
So Spermine was messing with P53's usual activity.
It was. And when they knocked down P53 in the macrophages, it reduced the Spermine-induced increase in pro-tumor cytokines, and it also reduced the expression of TDG.
So P53 was somehow involved in boosting TDG levels.
It was. And they confirmed this using a technique called chromatin immunoprecipitation, or ChIP.
ChIP, what's that?
It's a technique that lets you see which proteins are bound to specific regions of DNA.
Okay.
And they found that P53 was directly binding to the promoter region of the TDG gene.
The promoter region.
It's the region of DNA that controls the expression of a gene.
So P53 was basically turning up the volume on the TDG gene.
Exactly. It's a cascade of events. Spermine activates P53, which then boosts TDG, which leads to changes in DNA methylation and ultimately promotes the pro-tumor state in macrophages.
It's like a domino effect.
It is.
So to recap, we've got cancer cells secreting arginine, macrophages taking it up and converting it to spermine, spermine activating P53, which then increases TDG, leading to less DNA methylation and more PPARG activity, ultimately resulting in pro-tumor macrophages that suppress T cells. It's a pretty complex pathway, but it's fascinating how it all fits together.
It is.
But did they actually test any of this in living organisms? Like, did they try targeting any of these components in animals?
They did. They used a couple of different approaches. First, they used what are called immune-humanized mice.
Immune-humanized mice, what are those?
They're mice that have been engineered to have a human-like immune system.
So they're basically little avatars for studying human immune responses.
Exactly. And they found that knocking down TDG in the macrophages of these mice inhibited tumor growth, both under normal conditions and even when they gave the mice extra spermine.
So even when spermine levels were high, reducing TDG still had an effect.
It did. And this reduction in tumor growth was associated with fewer pro-tumor macrophages within the tumor and more CD8 plus T cells infiltrating the tumor.
So it was shifting the balance back in favor of the immune system.
It was. And in a separate experiment, they treated mice with normal immune systems with a drug called DFMO.
DFMO, that's...
It stands for defluoromethylornithine.
Okay, and what does DFMO do?
It's a drug that inhibits the production of polyamines.
So it's basically cutting off the supply of spermine?
Exactly.
Yeah.
And they found that treating mice with DFMO significantly slowed down tumor growth, but only in mice with functioning immune systems.
So again, it points to the importance of the immune system in this whole process.
It does. And the tumors in the DFMO-treated mice also had fewer pro-tumor macrophages and more CD8 plus T cells present.
So both knocking down TDG and blocking polyamine synthesis seem to have similar effects.
They did. They both tipped the balance in favor of the immune system.
It's really encouraging to see that targeting these components of the pathway can actually have an impact on tumor growth in living organisms.
It is. It opens up some really promising avenues for developing new treatments.
But before we get too carried away, were there any limitations to this study that the researchers themselves pointed out?
Yes, they did acknowledge a couple of things. First, they focused specifically on macrophages in this study, but they mentioned that other types of immune suppressive cells, like regulatory T cells and myeloid-derived suppressor cells, might also be involved in this process and warrant further investigation.
So there might be other players in this game that we haven't uncovered yet.
It's possible. And second, this study focused specifically on breast cancer, and it's possible that the specific metabolic interactions could differ in other types of cancer.
So more research is definitely needed.
Absolutely.
But even with those limitations, this study is a really significant step forward in understanding how cancer cells manipulate their environment to promote their own growth. So let's zoom out for a second and try to summarize the key takeaways from this research. What are the big picture messages here?
Well, the study provides really strong evidence that breast cancer cells actively manipulate arginine metabolism to promote their growth. They essentially convert macrophages into pro-tumor cells that suppress the rest of the immune system. And this whole process is fueled by the arginine produced by the cancer cells.
And that arginine leads to the production of polyamines, with spermine being a key player in driving those pro-tumor effects.
Right. Spermine triggers epigenetic changes through the enzyme TDG, which increases the activity of PPRG. And that ultimately leads to macrophages that suppress the activity of anti-tumor T cells.
It's like a chain reaction of sabotage.
It is. It's a really sophisticated way that cancer hijacks normal biological processes to its own advantage.
And knowing all this, what are the implications for treating cancer? Like, where do we go from here?
The therapeutic potential here is huge.
Yeah.
Targeting this arginine polyamine TDG axis could be a really effective way to fight cancer.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.